Thank you everybody for participating in this seminar. I am truly honored to being able to being the last speaker of this outstanding seminar Adiseo put together on such important topic. I have to thank especially to Damien, to Julie, and to Olga for inviting me. It's been a great experience for me trying to put together this presentation for you. I have given full rights for Adiseo to completely distribute this presentation to all the attendees that wants to keep it, to have it. It's your presentation. It's everybody's presentation now. And I would like to start this presentation asking a question: what is life? And it is a very difficult questions, even with all the technology that we currently have. We humans have not been able to create life out of nothing. Perhaps the molecule that it's the magic molecule that differentiates living organisms from non-living organisms are enzymes. And the way enzymes work is through using quantum mechanics laws, particularly quantum tunneling. Quantum tunneling is the way that explains everything from photosynthesis to birds' migration. And if you are really interested in this fascinating topic, I highly recommend a couple of books to you: Life on the Edge and the Physics of God. I assure you that your life will never be the same after reading these two incredible books. The way most of the presentation is focused on precisely enzymes. In the summer of 1960, an outbreak of an unknown disease killed 100,000 turkeys in 500 cases. This was named Turkey X disease to this mysterious disease. After investigation, it was finally traced to mycotoxins in groundnut meal feed imported from Brazil by the British cargo ship Rossetti. The feed was known to contain a compound that could cause cancer. And since then, over 500 mycotoxins has been identified. And these are secondary metabolites that are able to affect and have toxic effect in all forms of life in Earth. To try to understand mycotoxicology, today we know that these complex toxins are able to cause profound changes in the cells of living organisms, affecting the metabolome, the proteome, genome, and transcriptome. Basically, every single point of physiology is affected by these cells and by these toxins, affecting different organ functions, immunity, digestion, reproduction, and in general, the health of the individual, regardless if it's a eukaryote or prokaryote organism, plant or fungi. All animals, including all forms of life, are affected some way, somehow, by these toxins. Of course, the damage to the cells by these compounds depend on several factors, like the concentration of the toxins, the duration of the exposure at the farm level, how hygiene, humidity, ventilation, the management is, of course, the species of the animal, the strain, the age, the sex, the nutritional health status. There are many factors that will affect and interfere with the severity of this disease. We know now that the several animal species are more susceptible to some mycotoxins more than other animals. Particularly, turkeys are extremely sensitive to aflatoxins, but as you can see, pigs are from those 500 over 500 mycotoxins that have been discovered. They are grouped in 6 groups. And pigs are extremely sensitive to pretty much most of these compounds. We now, this is a very interesting paper that was recently published and explains the susceptibility in turkeys for aflatoxin. And these authors found that this susceptibility is associated with the inability of the hepatic glutathione transference to detoxify the reactive electrophilic metabolites of AFBO, or aflatoxin B1,8,9 epoxide, which is the extremely toxic compound for the hepatocytes in the animals. The cytochrome P450 superfamily of enzymes are mainly concentrated in the liver. And it's through this superfamily of enzymes that this important organ, the liver, particularly in animals, are able to detoxify and metabolize a lot of compounds, hydrolyzing, oxidating, and through these changes, through these enzymatic changes, are able to change and modify many of the toxic compounds that are found on the diet, produced by bacteria, fungi, parasites, or in infections, if there is injury or any other chemical exposure. This P450 cytochrome enzymes are able to detoxify a lot of compounds. In the case of aflatoxins, though, this is going to modify aflatoxins in a way that is going to make them even more toxic. But then there are other enzymes that are going to be able to degrade those oxidative compounds in different ways, as we're going to see. Of course, this interesting paper has been shown that masked mycotoxins are starting to be biodegraded by the host since the saliva. At the upper gastrointestinal level, from the saliva, using saliva, gastric juices, or duodenal juices, or bile, there is a beginning, and these investigators have been shown that there is some hydrolysis. But interestingly, we can see that is the microbiota through fecal fermentation in the lower gastrointestinal tract, where these masked mycotoxins have been able to be hydrolyzed over 97%, depending of the toxic compound they were looking. But it's very interesting to see that the interactions between mycotoxins and microbiota may also explain part of the differences in the susceptibility to mycotoxins among the animal species. We see that isolation of microbes present in the gut, responsible for the biotransformation for mycotoxins into less toxic metabolites and for binding mycotoxins, these findings led to the development of probiotics, enzymes, cell extracts that are currently used to prevent and reduce mycotoxic toxicity in animals. More recently, bio-inactivation of mycotoxins into less toxic compounds, notably through the hydrolysis of masked mycotoxins by enzymes, reveal that the health benefits of the gut microbiota or mycotoxins through the enzymes that they produce can vary strongly depending on the mycotoxins and the microbe that is involved. Mycotoxin research can be divided in three main areas of research. We can look at the cellular targets of the mycotoxins. This is in the epithelial cells, the goblet cells, and neurons, particularly in the gut-associated nervous system, which is considered today as the second brain. The enteric nervous system contains more neurons, over 100 million neurons, more neurons that are present in the whole peripheral nervous system, and mycotoxins can have a profound effect on these neurons in this important organ that is the intestine. We can look at the markers, look at oxidative stress and permeability, and we're going to see that this is one of the most negative effects that mycotoxins can have on gut integrity, increased gut permeability through oxidative stress, and of course, looking at the effects of the microbiota. Due to the direct toxic effects of mycotoxins on prokaryotes, and not only eukaryotes cells, but also prokaryotes, it's been known that mycotoxins can have profound changes in gut microbiota composition. And we can see, it's been shown that these changes can be evaluated not only at phylum level, but also genus, species, and species of these important microbiota that is in the GI tract, in the gastrointestinal tract. These changes of this toxic effect produced by mycotoxins on the microbiota, on the intestinal microbiota, can cause profound changes in this fragile ecologic system, modifying the eubiosis. This is the normal composition of beneficial bacteria to dysbiosis. By these changes, these dysbioses are associated with profound inflammatory effects in the GI tract. And we can see that any kind of stress, any kind of stress, not only at nutritional level by contamination of mycotoxins, we can see that the nutrient density, the compositions, if there are enzymes, the ingredients of the diets, but regardless, any stress can be environmental through heat stress, for example, ammonia, high density, can be produced by biologic stress, virus, bacteria, oncoparasites, could be psychological, emotional, any kind of stress, regardless of its origin, is going to have a profound impact on this fragile ecosystem in the GI tract, the microbiota, producing this bacteriosis. When we have a change in the normal profiles of beneficial bacteria in the gut, they are going to have profound effects in the health of the animals. Several years ago, Time magazine dedicated special issue to what they call the secret killer. And the secret killer was, or is identified as inflammation. It's been recognized that inflammation is associated with heart attacks, all types of cancer, Alzheimer, and many other diseases. The way this is working, and it's fascinating because here we need to remember and understand that about 80% of the immune system's cells, innate or acquired, are located in the GI tract, in the gastrointestinal tract. The gut is the most is one of the most important organs where we have high number of these immune cells, and the reason is clear. The GI tract is responsible for tolerating the food and the water the animals we absorb for our necessities, but at the same time it has to block the entrance, isolate the external environment from the internal environment and from all the bacteria that are located in the GI tract, the microbiota, or any pathogens that we can consume during the ingestion of food or water. So it's been considered that about 90% of all diseases, regardless of its origin, could be metabolic disease, autoimmune disease, any kind of cancer, enters in the GI tract. Its origin has direct influence by the inflammation in the gastrointestinal tract. And this is the association of knowing that there is such high number of immune cells in the GI tract because if there is gut inflammation, there's going to be tremendous repercussions. Inflammation has been associated with so many diseases. 90% of most of the diseases have an origin through gut inflammation. And the way this is working is because if we have gut inflammation, there's going to be disruption of very important tight junction proteins that are keeping the enterocytes together, isolating the external environment, the lumen, from the internal environment. When we have this disruption of the tight junction proteins, we're going to have what is known as leaky gut. And leaky gut then is going to have the penetration of antigens, bacteria, and many other negative stuff to the blood, causing systemic chronic inflammation. And that's how these inflammatory effects in the intestine have profound effects on all the cells of the organ, of the system. So when we have inflammation in the GI tract, there are not only negative consequences in the permeability of this important organ. It's been demonstrated that Salmonella, for example, benefits from intestinal inflammation. It's been shown by several investigators because, well, and in this case, not only Salmonella, but many enteropathogenic bacteria, gram-negative bacteria that have LPS in their cell wall. LPS, lipopolysaccharides, is very, very antigenic and induce inflammation in the GI tract. This compound, by inducing inflammation, is going to have very fascinating effects that benefits bacteria like Salmonella or E. coli or Campylobacter, because through the process of inflammation, we're going to have the secretion of several reoxide oxygen species. Oxygen is going to be that is produced by heterophils, by macrophages, trying to reduce it's a mechanism of innate cells to kill bacteria, and it's very effective. Oxygen reactive species are extremely toxic. Unfortunately, they are not toxic for bacteria or other microorganisms, but they are also toxic for the cells itself. But in this case, when we have inflammation that can be induced by a mycotoxin or a Salmonella, we're going to have this increase of oxygen reactive species that in the intestine are going to be, this excess of oxygen is going to join to thiosulfate, producing tetrationate in situ in the intestine. And for those of you that are not very familiar with tetrationate, well, tetrationate, it's been used by microbiologists in broth. It's to have tetrationate broth is a selective media to grow Salmonella when we are trying to do diagnostic of this pathogen from any organ that we are looking, or any tissue. So it's a selective media for growing Salmonella in vitro. Imagine what this bacteria has been able to do, produce tetrationate in the gut to increase the chances of the favor this growth of this bacteria. Tetrationate is also very toxic for many of the other beneficial bacteria. So that's how Salmonella uses these inflammation processes to benefit itself. And of course, other pathogens like Emeria. Emeria, the damage that Emeria parasites cause on the GI tract induce also severe inflammation and profound changes in the microbiota diversity. And it's been shown that Salmonella benefits from Coccidia infections through the inflammation that they produce. But mycotoxins basically is doing similar effects. There are other studies, several studies that have shown that mycotoxin also favor Coccidia parasites to induce, when we have the association of mycotoxins, Coccidia parasites also benefit from this, this damage in the intestinal mucosa. It is also known that mycotoxins are able to produce profound negative changes, toxic effect, not only in the enterocytes, but also in the goblet cells. The goblet cells are those cells that are in the GI tract and in many mucosas, genitourinary, respiratory tract, all the mucosas have goblet cells. And these are the cells that are in charge of producing mucus. And it's been shown that mycotoxins are able to negatively impact the these important cells that produce mucus. So there is we can see that the biological effects that mycotoxin are very profound, not only affecting and being able to destroy enterocytes, but also goblet cells and neurons. And this is a way that is going to have profound changes in gut physiology. Also, very important, very important is the effects that they have on the microbiota. All this inflammation by direct or indirect effects of mycotoxins are going to have tremendous impact on the microbiota, producing dysbiosis. When we have dysbiosis, we have more inflammation, and more inflammation is going to have more leaky gut, more bacterial translocation. So you can see the mycotoxins have negative effects, not only directly, for example, on the hepatocytes. Aflatoxins, by destroying hepatocytes, is very toxic, very negative, just like alcohol. And that is the problem of chronic alcoholism in humans. For example, the alcohols, just like mycotoxins, not only are toxic, very toxic for the hepatocytes, but also for the enterocytes, increasing gut permeability, this leaky gut syndrome, causing gut inflammation, which is going to have a profound effect, causing systemic inflammation. And chronic systemic inflammation is going to have profound changes in all the cells of the organism. Obesity, for example, obesity is a state of chronic inflammation too. And obesity has many effects, negative effects, in all the organs, all the tissues of individuals through this chronic systemic inflammation. So, what are the strategies that we have to reduce mycotoxin damage? Well, we need to understand the strategies to detoxify and repair the organs that are affected are one of the most important mechanisms that we have in terms of biology. How we can bio-inactivate mycotoxin is through the enzymes. And in this case, produced by microbes, bacteria, by yeast, and enzymes. This magic molecule can have profound beneficial effects through the enzymes that the microbiota can produce, or the host itself. So we can see, and it's been shown, and that several investigators have been able to isolate bacteria from the soil, some environmental samples, and many of these bacteria have shown that to produce different enzymes that are able to bio-inactivate the mycotoxins. Time is my limitation for this presentation. I wish I could have more time to get into the details of these fascinating articles that have been able to show how different bacteria isolated from soil have on the bio-inactivation properties for mycotoxins. And there have been several. You can always email me. I can share these articles. You can download them themselves. Most of them are open access. And the objective of this talk is to show you that there are so many bacteria, unknown and known, that are out there that have been able to bio-inactivate mycotoxins through enzymatic fascinating enzymes. And there are also many other studies that have shown many scientists have been able to isolate bacteria from gastrointestinal tract of chickens, and these bacteria have been able to bio-inactivate several mycotoxins that are affecting animals. Pigs have been also used, bacteria isolated from the normal gut microflora for pigs have been able to show by several investigators that are also important compounds to bio-inactivate mycotoxins through the enzymes that these microbiota organisms have. We're talking about isolation of specific bacteria, and these bacteria have been isolated from rumen fluid also. Rumen protozoas, rumen bacteria that produce these enzymes that are being able to detoxification of the mycotoxins. It is fascinating that bacteria, the different ways bacteria are able to reduce the severity of mycotoxin in animals. Perhaps some of the most studied mechanisms are the binding. And the binding of, for example, lactobacillus. Several lactobacillus have the ability to absorb mycotoxins in the GI tract, and the binding is so strong, it's irreversible. And it's been shown that these mechanisms involve several compounds that are on the cell membrane of lactobacillus, and the binding involves hydrogen bonds and van der Waals forces. And it's so strong that this binding, when lactobacillus bind mycotoxins, then mycotoxins are excreted in the feces of the animals. So the mechanism of the bindings are at different molecular structures that lactobacillus have in the cell membrane. And this has been studied for many, many years. But more recently, it was also shown that lactobacillus produce several enzymes that directly can also have the ability to produce biodegradation of enzymes. And not only the enzymes that they can produce, but the metabolites that they produce are able to also reduce or bio-inactivate several mycotoxins. This particular article, for those that are interested in studying in more deep details the different ways biological detoxification of mycotoxins by not only lactobacillus, but in general lactic acid bacteria have to reduce the severity of this disease. Knowing this basis, we can see how and why bacteria has been able to use to being one of the most important bio-inactivation tools that we can have in our hands to reduce the severity of mycotoxins. And the strategies that we have is not only, of course, by the ingredients of the diet, the quality of the ingredients that we are providing to ourselves or our animals, but we can also use probiotics or prebiotics, which prebiotics are basically feed for increasing beneficial bacteria. We can look at the symbiotics, which is the combination of probiotics and prebiotics, and we can associate this like providing the seed and the fertilizer together. So we are giving the beneficial live bacteria and the food that they have to increase their numbers in the GI tract and have beneficial effects, profound beneficial effects, not only by bio-inactivation of mycotoxins, but also changing the physiology of the animal, because the microbiota that we have in the GI tract of all animals represents our second genome. The microbiota that we have in our gut, it's overpassed the number 10 to 1. We have 10 bacteria for every somatic cell in all animals, regardless if it's a bee, a pig, or a human. The proportion is a proportion estimated 10 to 1. And this number, this association, this number 10 to 1 is not only given by the number of bacteria versus cells, but also by the genes. So the microbiome, the genes of the microbiota, it's also overpassing the genome of animals. It's been estimated that the microbiota genes are about 300,000 genes. So you can see that the association, we live on Earth also on all animal species depend on this fragile microbiota. That's why, and they have profound effect on the physiology. We could talk hours about all the beneficial changes that beneficial bacteria do on the immune system, the enteric nervous system, endocrine system, all the physiology, the health of the animal depends on this fragile ecosystem. So by providing prebiotics, or the combination in symbiotics, we can have this beneficial effect of this bacteria. Phytobiotics are another compound that we have, another tool to reduce the severity of mycotoxin, and of course, the use of enzymes. For many years, it's been known the physical ability of yeast cell wall and yeast cell constituents to bind mycotoxins. Mycotoxin binding can be enhanced greatly by the use of yeast cells or the components of the yeast cells, mainly beta-glucans, beta-glucans and mannan oligosaccharides, instead of the whole cells. The fact that dead cells do not lose their binding ability shows that the interaction of such molecules to bind mycotoxins by addition is rather by these wall components, rather by covalent binding or by metabolisms or other like enzymes in living organisms. So we have been also in our laboratories being able to look at different phytobiotics and interestingly, very recently, we have shown, and we're working with lettuce. Lettuce, some of components of lettuce are able to have incredible properties to bind mycotoxins, even stronger than the most common adsorbents known out there in terms of commercial application. And this is we're currently working on these fascinating compounds with another group of scientists at the National University in Mexico, UNAM. But of course, of course, enzymes. Enzymes, it's the key. The key of the biodegradation, bio-inactivation of mycotoxins is done by different enzymes. The detoxification, the changes in the structure of these toxic compounds is thanks to the production of the enzymes. And in several years ago, we were looking on isolation, screening, and identification of bacillus as direct fed microbial candidates to detoxify aflatoxin B1. We found one Bacillus subtilis that in vitro show the activity to biodegrade aflatoxin B1. Unfortunately, when we test this that direct fed microbial in vivo in the chickens, and we gave these chickens a diet contaminated with aflatoxin B1, we didn't see any effect to reduce the severity of the disease. However, we continue our research, isolating other bacillus candidates that were able to produce exogenous high concentration of lipase, phytase, proteases, and several carbohydrates. And by the selection of these in vitro properties of these bacillus, we create another direct fed microbial that when we test in chickens that they receive a diet contaminated with aflatoxin B1, we found, and this was a very interesting manuscript that I recommended to you. Give it a read. It's incredible because we were able to see that this direct fed microbial was able to improve the performance, the immunological status, and the serum biochemical parameters of the chickens that received this direct fed microbial compared with control birds. And this was very, very interesting. Curcumin. Curcumin is another phytobiotic compound that it's out there with incredible properties. Perhaps the most important properties of curcumin are the antioxidant activity, the inhibition of aflatoxin biotransformation, because in the case of cytochrome P450, this activity of this powerful enzyme produce more toxic effects, the AFBO. Well, there is this indication that curcumin can also reduce this bioactivation and reduce the more potent toxic effect of aflatoxin that way. And of course, by immunomodulator effects. So I wish I had more time to get into deeper, profound effects of the benefits of curcumin. I just wanna show you that we've been testing and evaluating several cellulosic polymers and curcumin to reduce aflatoxin B1 effect on performance and immunological parameters in broiler chickens. And we have seen incredible benefits, like has been shown by many other scientists. More recently this year, we just published this article where we were able to show the effect of curcumin and copper acetate against salmonella infections on improving the intestinal permeability and microbial composition in broiler chickens. So, what about essential oils? Essential oils like thyme, and you can Google mycotoxins and essential oils, and you're going to see an incredible number of publications out there showing that essential oil can partially restore the negative effects of mycotoxins on several animals, but the way they are able to produce and reduce these effects are, of course, essential oils have antioxidant, antifungal, antibacterial, immune modulated properties, but the way essential oils ameliorate the negative effect of mycotoxins is by their antioxidant properties. You already were able to have and listen a specific lecture on the oxidative stress negative effects of mycotoxins on the cell. So that's how essential oils can help in increasing the overall health status of the birds by their antioxidant, anti inflammatory properties that they have, and also increasing the immune system. So with this, I thank you very much for your attention. And let me tell you and assure you that you can always contact me by email. And here at the University of Arkansas, you have a friend. And if I can do anything for any person out there, please contact me. I'll be more than happy to try to do whatever I can to help and answer your questions. Thank you very much for your attention.
Okay, thank you. Great presentation. I hope everybody enjoyed. So we now have about 15 minutes to answer your questions. If you want to type them on the chat area on this right side of your screen, I'll be glad to read them to Dr. Julia Dvorska. And I'll go ahead and start with the first one here. Do antibiotics reduce the ability of hosts to cope with mycotoxins?
Yes, thank you, Guillermo. So as we heard from the Emma's presentation, the microbiota is like the second brain and very important for the host at all. Not just speaking about the mycotoxin degradation, but if we're using the antibiotics and they can kill microbiota, so we are like reducing the natural ability of microflora to defend against mycotoxins, to produce metabolites, to produce enzymes, which can be helpful to biodegrade mycotoxins. So yes, I think it's very important. And if we speak about the ruminants, for example, some of mycotoxins like penicillium mycotoxins, they are important for silage. They can have antibiotic effect already. So if we have mycotoxins and feed it to ruminants, and they can already decrease the ability of microbiota of rumen microflora to degrade mycotoxins. Yes, and we know that, for example, for ruminants, microbiota plays really important role in degradation of trichotigens like DON, T2 toxin, and ochratoxin A can be degraded by healthy microflora to 100%. But if we have some decrease in microflora or have antibiotics or penicillium mycotoxins, well, we have negative impact on the microbiota, and also some conditions like acidosis and can switch microbiota. Like during this presentation, you remember Dr. Memo had shown that we have like balance of good and bad microbiota always. And in this case, we have like this balance is already going down with level of pathogens increasing. So yeah, it's a very important topic. I agree.
Okay, thank you. Next question here. Do the microbiota is the only responsible tool for mycotoxin destruction in the animal?
Yes, microbiota is one of the important, but we also have liver cells, hepatocytes, and again, as we've seen today during this presentation, that they can produce enzymes, and enzymes can degrade mycotoxins. Also, enterocytes can be the host cells which can degrade mycotoxins. Phagocytes sometimes can be also effective against mycotoxins. So we are not speaking about just about the microbiota. Even microbiota is number one, but still we have other cells on host which can help us. For example, liver cells. You know that liver can degrade, for example, DON very effectively in pigs to DON3 glucuronate and in poultry to DON3 sulfate. So it's liver cells which are degrading or biotransforming the mycotoxins.
Perfect. Thank you. Next question here. What is the best way to combine probiotics and prebiotics to have maximum effect? Take them at the same time or alternately?
Mhm. Yeah. Again, we have heard today about symbiotics, like they're together, but we need to know what is the aim of the usage of these probiotics and prebiotics. For example, if we had antibiotics before, like antibiotics treatment, and we need to help microbiota to seed, like he mentioned during the doctor mentioned during this presentation, like to seed microbiota, then we use probiotics. But if we want to boost the host microbiota, which already present in the gut, we will need to use prebiotics like yeast or yeast extract or whatever to have a fertilizer or to feed microbiota. So it's dependent on the aim, what aim we have. Yeah. So
Okay, thank you. Moving forward here to the next one. Would a binder be the best approach to help on the host microbiota to respond to a mycotoxin challenge?
that's interesting question. Well, binding is very well because it's like well, first of all, we need to try to eliminate as much of mycotoxin as possible. For example, we know that, for example, aflatoxin B1 is very well absorbed by normal clays, and it can be absorbed in vivo 80%, even in vitro 100%. And yeah, we need to try to eliminate as much as possible. For example, we're speaking about aflatoxin or ergots, alkaloids. They are very well absorbed by clays, or like yeast cell walls or yeast, some amount of zearalenone also can be absorbed and eliminated. And then, but we need to understand that we cannot absorb 100%, even in case of aflatoxin B1. It's because it's very fast absorbed, and just binder cannot do it like very fast and in time. So we have like 20% of in case of aflatoxin B1 adsorbed, and now we need to help microbiota, to help liver, to help microbiota to biodegrade or to cope with that amount we have already in the gut. And the same with other mycotoxins. Some of them, you know, that, for example, difficult to absorb DON, really difficult, but even if someone can absorb some DON and take it through all the gut to the low part of gut, and then it can be, because adsorbed is doesn't mean that it's that it's not active. In most of cases, the mycotoxin still active in binders, and they goes to the lower part of gut and can damage the enterocytes, because for DON, the target organ is gut. So not really good idea to absorb DON. Better to help the body to help host cells to bio-inactivate. Because, yeah, as I mentioned already, the liver can biodegrade it very well. And most important, it's absorbed very fast. It's absorbed very fast and already in stomach. So, so in this case, yes, we can. First of all, we can bind if we can bind something, and then help microbiota, help microflora, host cells to deal with this in natural way, like it's always done in nature. You remember, for example, or you have seen like wild swine or swine when they're in open spaces somewhere in the nature, they can eat soil. They eat soil very often. And soil microbiota, as we've seen during today's presentation, soil microbiota can degrade toxins very well. So it's like natural protection and natural mechanism, which can be also boosted by using some other products like you mentioned, binders and some yeast or whatever. Yeah.
Okay, thank you. You mentioned a little bit about ruminants already, so there's a question here about it.
Mm-hmm.
in view of the extensive role of the microbiota in defense against mycotoxins, what is the situation in ruminants?
Yes, the situation with ruminant is like all the same, because we, for many years, we thought that the ruminants are not susceptible to mycotoxins, and it's not like a problem at all for ruminants, mycotoxins, since they have a rumen microflora, and that's true. That's true. The rumen microbiota, like bacteria and protozoa, yeasts, they all working together and can degrade mycotoxins successfully again, but to some extent, if you have healthy microflora, if we have like good conditions, no acidosis, everything is perfect, then we would expect that it's at least trichoticins and ochratoxin A will be degraded. But at the same time, in rumen microflora cannot degrade anything of aflatoxin, nothing, zero. And other mycotoxins like cyclopentazonic acid and many others, it's like zero biotransformation, meaning that even if we have microbiota, we need to take care of ruminant in this case, especially speaking about aflatoxin B1. And again, like in silage, we have a lot of penicillium mycotoxins, which can have negative effect on the rumen microbiota, on yeast and the protozoa and bacteria. So.
Okay, thank you. We have one final question here, saying enzymes were mentioned several times during the presentation. Should we add pure enzymes or just support a host bacteria to produce them?
Well, also very interesting questions. Well, my opinion is that the host microbiota can produce enzymes very successfully, and not just enzymes, also some other metabolites. Like we've seen during this presentation, lactobacillus example. Lactobacillus can, first of all, bind some zearalenone, and also bind and take it from the organism to the fishes, and also produce enzymes, lactobacillus, and produce another metabolites. And they can biodegrade or deactivate mycotoxins. But if you have external enzymes, well, it's also good, but we need to make sure that they are starting to work already in the stomach, already in the acidic conditions, because as I mentioned, for example, DON or zearalenone, they are very, or aflatoxin B1, they are really fast absorbed and already from the stomach. So if most of the external enzymes, they're starting to work in the gut, in the low gut, when pH is 6.5. And in this case, it's already too late to biodegrade mycotoxins, which are already absorbed in stomach. So if we have some enzymes and they are working already in stomach, well, good. But we need to make sure that. But still, I believe that the natural microbiota can be much more effective with producing not just enzymes and other metabolites, which can be also important and help to biodegrade.
Okay, thank you. I don't have any other question here in the chat, so I would like to thank everybody for joining us today. It was a great series with these 5 webinars. I think we could cover a lot of the main topics around mycotoxins and the challenges to animals, including bio-inactivations we had today, oxidative stress problems as we heard before, the binding of mycotoxins. So we could cover a lot of the different things the industry discuss on these topics. I appreciate Julia for taking the time to answer all the questions we had, and we will be in touch with you for new ideas, new ways to bring information to you in the industry, and we'll definitely have some very good things to present in the future. So thank you again, and I will give the floor to Mateus just to say a couple words to the team in South America. Thank you very much.
Obrigado, Guilherme. Thank you very much, and obrigado, amigos de América Latina. Gostaria somente de agradecer de verdade a oportunidade de ter vocês aqui com nós outros mais uma vez. E lembrar que, a partir da próxima semana, teremos todos os webinars, todos os episódios do MycoInfo disponíveis no Feed Channel. Espero que, caso vocês tenham perdido algum dos episódios, possam conectar para assistir novamente, porque é uma excelente oportunidade. Obrigado a todos. Buenos dias.
Okay, thanks a lot. Thank you. Bye-bye.
Bye-bye.